Our second speaker today is Associate Professor Brian Billups. Brian's lab, the Synaptic Mechanisms Laboratory, investigates how individual synapses in the central nervous system function and how they're modulated. He has put together a wonderful talk for today. How do neurons communicate? Thank you, Brian. Please take it away.
Hi, so let me work out sharing my screen. Okay, good morning, everyone. And I'd like to begin by acknowledging I'm sitting here in Canberra in the land of the Ngunnawal people and pay my respects to their elders, past, present, and emerging. So my talk today is about how neurons communicate. And as you'll know, this is part of our BrainTeaser 2021 program today. So you just heard a talk from John, and he described how the nervous system was built up and how these cells in your brain called neurons are structured something like this. You have a cell body and you have dendrites on the top, which is the word dendrite comes from the Latin for tree. So there's processes coming out like a tree, and this is where the inputs into a neuron come to, and then there is an output down the axon. And these neurons communicate via electrical signals. So electrical signals are going through the dendrites and out of the axon like electricity running down wires. Okay, and John showed you a picture of a real neuron here. And just for scale, I put a human hair here. This is roughly the width of a human hair. And notice that the cell body of a neuron is less than the width of a human hair. So these things are tiny. But some of their axons can be very, very long. They can stretch up to 1 meter away from the cell body. So although the cell bodies are tiny, they have a far-reaching processes. And as John mentioned then, these neurons are wired together in circuits, okay, where they join together. And where the electrical contact of one neuron is communicating with the second neuron, this is a place called the synapse. And this talk today I'm going to give you is going to be focused on the communication between neurons which occurs at that synapse. So as he introduced, there's an electrical signal coming down neuron 1 here, and there's a gap here between the two, and there's a chemical signal which crosses this gap and is turned into an electrical signal going down to the other neuron. Okay, so that's how these neurons are wired together in these networks. And this really is the essence of how your nervous system works. Everything that your nervous system does can be broken down into these neurons and these synaptic connections between those neurons. Everything from how your body moves, how you think, emotions, and memories, feelings, everything is coming down to those neurons and particularly those synapses. Now, this is a cartoon image, and it doesn't really do justice to how complicated the system is. But this is actually a real picture of neurons inside your brain. Now, this is a special mouse produced by these guys over 10 years ago now, and this is called the rainbow mouse. Now, the rainbow mouse is genetically engineered, so each of its neurons is a different color. And this is showing a photograph of part of the brain called the hippocampus. This is a tiny little bit in the middle that's responsible for encoding memories in your brain. Now, this whole section here is just over 1 millimeter across, but you can see within that there are thousands and thousands of these neurons here in the hippocampus. Right? The inputs are coming in here to the dendrites, and they're all intertwined and all packed together really, really densely that it's so hard to see an individual neuron. And then the outputs are all coming out here, and these are the bundle of axons coming out to other areas of the hippocampus. This is just part of the hippocampus. So you really get a sense from this picture of how densely packed these things are. And there's 100 billion neurons or so in the brain. That's as many stars as there are in the Milky Way. So when you look up at night and a dark night and you can see all those stars, that's just a fraction of the number of neurons that there are in your brain. But it's easier to think in terms of cartoons because you can get a sense then of how these neurons are joined together in a network, and where they communicate, these processes of these neurons actually come together and touch. And here I've just put a little cartoon, which is a section of the creation of Adam, drawn by or painted by Michelangelo on the roof of the Sistine Chapel. You can see this picture here. Now, Michelangelo was a keen anatomist, and he was very aware, he was aware of the structure of the brain. And it's thought that this picture here, this is God here creating Adam, and this represents the human brain. The structures and the lines are very clearly focused and located in the same place as some of the folds of the human brain. And this sash here is where one of the major blood vessels are. So scholars have interpreted this to be a representation of God giving intelligence to Adam, and they're communicating by reaching out their processes and touching, just like neurons communicate in the brain. However, what you notice is that they come very, very close, but they don't actually touch. And this is how synapses are in the brain. So I'm going to draw an analogy here between the hand between God on one side and Adam touching, and the synapse in the brain, where there's a little gap between our neurons where this chemical transmitter must jump across. Okay, now, obviously, I've drawn this analogy a bit far over a glass of wine last night. This is just what I came up with. Now, Michelangelo was very aware of the structure of the brain, but he'd no idea about synapses. Synapses are tiny. They're about a 4,000th of the size of a human brain. So he had no idea that these things existed. Even our very own John Eccles was skeptical about the existence of synapses. As late on as 1949, he believed neurons communicated by direct electrical transmission, just the electrical spread of signals down neurons in what's termed action potentials. And he got the Nobel Prize for discovering the mechanism of these action potentials, these electrical signals. But it was two of his colleagues who really spearheaded this idea. So Stephen Kuffler was Eccles' PhD student here in Australia. Bernard Katz worked with him in Australia before going on to work in the UK in London for many, many years. What these guys did is they put sharp electrodes inside of neurons, and they measured the communication between the neurons and the time it took. And what they realized was that there was a very tiny delay between the signal from one neuron to another neuron. And if it was purely an electrical spread down the wire, there wouldn't have been a delay. But because there was a tiny delay, less than 1,000th of a second, it caused them to propose that there was this chemical step in between the neuro communication. Okay, and advancing these ideas, Katz went on to get the Nobel Prize for work done in the 50s. It's awarded sometime later, but he did this work in the 50s, discovering that neurotransmitter was contained in little packets called vesicles. This is a chemical neurotransmitter that's released from one neuron and activates another. Now, this is remarkable because at the time, microscopes were not good enough to be able to actually see these structures. Sometime later, the electron microscope was developed, and then we could really see these synapses. So this is a very classic picture of a synapse in your brain. What you see here, this is the end of the axon from one neuron here. This is just the very tip of that axon. And that's communicating with the second neuron, which is down in this area here. So in there, we see lots of these mitochondria. Synapses have lots of mitochondria in because this is a very energy-intensive process. These are the powerhouse of the cell, producing the ATP for the energy driving this process. But what you'll also see are these little bubbles of vesicles. These are synaptic vesicles. They're little balloons of membrane which contain a chemical neurotransmitter. And they release that to activate the postsynaptic cell. And what you can clearly see is this gap in between the two cells running here. If you're lucky with taking the picture at just exactly the right time, what you can see is one of these vesicles merging with that membrane and releasing its contents out to the gap between the two cells here in real life. Okay, but it's a little easier to see in cartoons, so we'll go back to a cartoon here. Here's a neuron coming in with its electrical signal, some synapses here communicating with the output neuron. So if we zoom in, it looks a bit like this. This is our synapse, our gap between the neurons. The electrical signal comes down here. It can't directly activate here. There has to be this chemical signal to cross this gap. So this gap is called the synapse. This is the synaptic cleft. The neuron coming in is called the presynaptic neuron, before synapse. Then the neuron coming out we'll call the postsynaptic neuron, post meaning after the synapse. And inside the synapse then are these vesicles, these membrane-bound sacs which contain this neurotransmitter. Okay, and when the electrical signal comes down here, these vesicles merge with the presynaptic membrane, release that neurotransmitter, and activate the postsynaptic neuron. Okay, bringing all that together then, I'm going to show you a very short video just to give you a sense of how these neurons flashing their electrical signals down, and we have to thank Elon Musk's company Neuralink for this video. They're doing lots of research into how neuronal communication can be harnessed to drive machines. But it's a great video, so have a look at this video.
Your brain is composed of nearly 100 billion cells called neurons. Neurons come in many complex shapes, but generally they have a dendritic arbor, a cell body called a soma, and an axon. The neurons of your brain connect to form a large network through axon-dendrite junctions called synapses. At these connection points, neurons communicate with each other using chemical signals called neurotransmitters. Neurotransmitters are released from the end of an axon in response to an electrical spike called an action potential. When a cell receives enough of the right kind of neurotransmitter input, a chain reaction is triggered that causes an action potential to fire, and the neuron to in turn relay messages to its own downstream synapses. Action potentials produce an electric field that spreads from the neuron and can be detected by placing electrodes nearby, allowing recording of the information represented by a neuron.
Okay, it's a nice little animation, just putting all that into context and showing that communication in this cartoon form. Okay, moving on now to think about some more detail of how these synapses actually work. So here's another schematic synapse here. We have a presynaptic terminal, vesicles with neurotransmitter in them, and the postsynaptic side with a receptor for that neurotransmitter. An electrical signal comes into this synapse, and that causes the vesicle to merge with the membrane and release its neurotransmitter. Now, this is a very complex biochemical process of releasing the neurotransmitter vesicles here, merging these vesicles with the membrane. It's driven by little nanoscale machines, individual proteins which join together and cause that release process. And this is a little cartoon I have here showing how this occurs. Okay, so we know what these proteins are, and we know how these little machines really work together to provide the energy to merge those membranes and to release those vesicles. And this all happens very, very quickly. Thousands and thousands of times a second. Okay, so after the neurotransmitter is released, then it binds on to the postsynaptic membrane here, and that causes that electrical signal to carry on onto the postsynaptic cell. This is how most of the synapses in your brain work. One synapse excites the postsynaptic cell. And the neurotransmitter molecule would be the molecule glutamate. This is the most prevalent neurotransmitter in your brain. It's a simple chemical molecule. It's actually amino acid, one of the amino acids that go together to make proteins in your body. But on its own, in your brain, it's a neurotransmitter. And the structure is shown here: 4 carbon atoms, an amino and an acids group. Not all neurotransmitters excite, some inhibit. So the neurotransmitter GABA, gamma-aminobutyric acid, has a very similar structure to glutamate. When that's released from a synapse, that activates different receptors on the cells, and that opposes this excitation and causes inhibition. So a large postsynaptic neuron in your brain can have up to 1 million synapses going on, at least several 100,000 synapses on a typical brain. Some are glutamatergic, some are GABAergic. This postsynaptic neuron is performing a computation between all the pluses and all the minuses coming in to integrate all those signals to give its one output down its one axon to decide if it's firing or not. Okay, so this is how neurons are performing computational processes. Okay, now for any signaling system to work, obviously the signal is turned on, but the signal has to be reset, it has to be turned off. How does that occur? Well, after the neurotransmitter is bound to the receptor, it diffuses away and the receptor stops working. The vesicle is pulled empty back into the presynaptic terminal. You have to recycle this neurotransmitter to allow this to reoccur. This is done in many different ways, but one way this occurs is by taking the neurotransmitter back up into the presynaptic terminal with a special little pump molecule. Another of these special nanomolecular machines which uses energy and pumps neurotransmitter back into vesicles. We'll call this a reuptake transporter. Once the neurotransmitter is back inside the terminal here, there's another of these special pump molecules to get the neurotransmitter into the vesicles, ready to be released again. So bringing all that together then, our postsynaptic neuron down here has excitatory signals, it has inhibitory signals, it has different neurotransmitters, and it has recycling mechanisms for the neurotransmitters and getting the neurotransmitters into the vesicles. This is a very complex system. Why do we have synapses, you might think? Well, this is how the neurons can regulate their communication. There's many, many points of regulation here that the brain can use to make the communication stronger, weaker, faster, slower, modulate it in different ways. We can modulate the number of receptors. We can modulate the amount of transmitter. We can modulate how long the transmitter is around in the cleft, how it's recycled, and how it's produced. So that gives a very rich variety of ways which the neurons can change our communication in our brain. Now, there's different transmitters. I've just introduced you to glutamate and GABA. That's the main excitatory transmitter, the main inhibitory transmitter. But there's more than 100 different neurotransmitters inside your brain. Now, some of them excite and some inhibit, as I said, glutamate and GABA. Some of them are very fast and some of them are very slow. So glutamate is a very fast neurotransmitter. It's a neurotransmitter the cells in your eye are using to detect vision. Think how fast that's working. It's the neurotransmitter your ears are using to detect the sound I'm talking. So it's very fast signals going on and off. Serotonin is a slow neurotransmitter. Serotonin neurotransmission is responsible for your mood, for your happiness, also appetite, also regulating sleep. These things happen over minutes, hours, days, or even months. Okay? Serotonin is a slow neurotransmitter. Okay, with all these different neurotransmitters, all the different ways that the brain regulates neurotransmission, it's a very plastic system. It's a system that can be molded. It's a system that can be changed. And it's also the place where drugs that alter our brain are affecting. They're really affecting these synapses. And just for an example, to finish off, I'm going to talk about 2 classes of drugs that alter your neuronal communication. The first one I'm going to mention are anxiolytics. Now, an anxiolytic drug is one which reduces anxiety, it reduces stress. So such a drug, for example, would be diazepam, also known as Valium. Now, this works at the synapse by activating those inhibitory GABA receptors. So when our postsynaptic neuron here is integrating its excitatory signals and its inhibitory signals to give an overall level of activity. It's just in the balance that's giving a normal level of activity. Sometimes when things go wrong, the neuron can get slightly overexcited and you can get anxious. So a drug like diazepam comes in here and it increases the ability for GABA to activate these inhibitory receptors. That enhances the inhibition and just gradually, gently pushes that neuron more to an inhibitory state than an excitatory state. It's quite a gentle drug, and it does this in a very controlled way. Now, there are stronger drugs which activate the GABAergic system, such as some anesthetics work, and that can completely shut your brain down by overexciting the GABA system. So this is a way that we can modulate communication in our brain by enhancing a receptor. The second example I'm going to give you then is antidepressants. Now, I mentioned that serotonin was the neurotransmitter that's responsible for happiness. Now, if you don't have enough serotonin, you can get depressed. People who have depression have reduced amounts of serotonin in their brain, generally. And one class of antidepressant drugs, there are others, but one, the main class, are these specific serotonin reuptake inhibitors, or SSRIs, such as the drug fluoxetine, known as Prozac, or the drug escitalopram, which is probably one of the most widely prescribed drugs in Australia, Lexapro, and it's an SSRI and it works on serotonin reuptake. So this is a synapse that's releasing serotonin here. When the serotonin has finished activating the postsynaptic cell, the serotonin reuptake transporter sucks it back up into the presynaptic terminal and turns off the signal. Our serotonin reuptake inhibitor then blocks this. So as more serotonin is released, there's more of it around to activate the postsynaptic cell, and our mood increases. Now, this is a very generalistic way of how SSRIs work, but it gives you the broad idea of how you can take a drug which interacts with your neuronal communication and has an effect on a particular neurotransmitter, which has a particular behavioral effect in your body. So as I said then, this is a complex system with many, many points of regulation. Now, we know some things about the neurotransmitters in our brain. We know some things about how this synaptic communication works, but there's an awful lot we don't know. And it's an ongoing area of research in many labs across the world, and also in labs in Australia and labs here in Canberra in the Eccles Institute of Neuroscience. We're studying how these neurotransmitters are released, how they're processed, how they're recycled, and how that can affect behavior in your brain. I'm going to leave it there for now, and I'll be very happy to take any questions. Thank you.
Thank you, Brian, for such an interesting talk and explaining all the mechanisms in such detail for everyone to understand. We've got a lot of questions. Maybe we could start with Anjali. Are there different mechanisms that regulate what neurotransmitter is used in synaptic communication?
The different mechanisms that regulate what neurotransmitter is used in synaptic communication. Well, yes, I mean, different neurotransmitters are released by different cells. There used to be a thing called Dale's principle, and Dale's principle, named after Henry Dale, who got a Nobel Prize for understanding neurotransmission in the 1930s, and that basically said that each neuron releases one type of neurotransmitter. So it gets lots of inputs from different neurotransmitters, excitation from glutamate, inhibition from GABA, regulation from serotonin, but it only releases one type from its presynaptic terminals. So if it's an excitatory neuron, it would release glutamate. If it's an inhibitory neuron, it would release GABA. If it's a modulatory neuron, it would release serotonin or dopamine, etc. Not necessarily strictly true now, because it's known some neurons can multiply release different neurotransmitters, but as a general principle, each neuron type releases one neurotransmitter to activate its neighbors. So it's sensitive to a range of neurotransmitters. It releases only one. The release mechanisms of the different neurotransmitters may be slightly different. So glutamate is a fast neurotransmitter, and that releases, you have these vesicles and they're on the membrane, and they can actually release by quickly opening and quickly closing, and they release neurotransmitter. The slower neurotransmitters can release by fully fusing with the membrane, and that's a bit slower. Some neurotransmitters are peptides, such as the endorphin neurotransmitters. And they tend to be in bigger vesicles, and they're released more slowly, and those vesicles are recycled in a different way. And those peptides aren't really recycled in the same way, they're just broken down, and more peptides are synthesized by the neuron. So there's differences there between the fast neurotransmitters like glutamate and the slower ones and the bigger ones like the peptides.
Thanks, Brian. I've got one follow-up question. So when there's all these cells communicating with each other at once, and also close together and releasing neurotransmitters at the same time, do cells ever make mistakes? And if they do make mistakes, what do they do to cope?
Whether it's a mistake or not, you're not really sure. But what I've shown you is a rather simplified view where one presynaptic neuron synapses with one postsynaptic cell, and you've got your neurotransmitter in the middle, and that just communication is there. But if you overstimulate and you get lots and lots of neurotransmitter here, it's what's called spillover, and that leaks out from that synapse, and it affects the synapses next to it. And it's thought that that isn't necessarily how these things are supposed to work, because for the most computing power, you want every synapse to be independent, but too much excitation, then this spillover occurs, and the neurotransmitters get to where they maybe shouldn't. And too much neurotransmitter can actually be toxic. If you put too much glutamate on a cell, the cells can die quite quickly. So it's very important that you quickly take it back up with those pumps back into cells. If that process breaks down, then that can cause neurons to die.
Yeah, that's very interesting, because the next question was what happens if excess serotonin is released by Shukla, which I think you covered in your presentation as well.
Yes, excess serotonin. It can be dangerous. You can get serotonin toxicity. It can massively increase your heart rate. It can increase your temperature too high. There are certain drugs of abuse which increase serotonin, particularly MDMA, ecstasy, and you can get what's called serotonin shock if you take too much. And you try and drink lots and lots of water to bring your body temperature down. If it gets out of hand, it doesn't work. But excess serotonin can be not good for you. Yeah.
And I think we have a question from Dave, which is more about general neuroscience. When forming memories, is one memory stored in one neuron, or is it partially stored in a number of neurons?
Yeah, so there's a thing called the grandmother neuron. I don't know if you've heard about the grandmother neuron, but the idea is that, you know, you have all these memories stored in your brain, and if one neuron stored one thing, then you'd have one neuron in your brain that was responsible for the memory of your grandmother. The problem there would be if that one neuron died, you would forget about your grandmother, and you wouldn't forget anything else, you'd just forget that one person, right? And as you get older, neurons die. And that's the sort of evidence that it's not one neuron, one memory, right? So you've got a big network of interconnected synapses. One neuron could have hundreds of thousands of synapses coming on. And the memory is stored in those synapses, but it's distributed over the whole network. And there's a whole field called neural networks where we study how these networks can store memories. And you can do this from a computational point of view and sort of neuron modeling and big computing power. And a good neuronal network is quite robust, and you can lose bits of it and you don't lose the entire memory. Some bits of the memory might become a bit blurred, and if you don't regenerate and renew the memory now and again, it can gradually fade. But because that memory is distributed in the storage in synapses over the network, then it's more robust to the grandmother cell problem where you don't instantly lose one very specific part of your memory.
Right. Thank you. I think we have one last question if you have time.
Sure.
Do you take into consideration the neurovascular unit's role in synaptic activity? And Aidan's asking if you investigate astrocytes, microglia, and capillaries in addition.
Okay, well, in a short talk, we haven't had time to talk about everything about the nervous system, but there are neurons, but there are also other cells in the nervous system, and glial cells, which are support cells for the neurons. There's different types of glial cells in your brain, and as you say, astrocytes and microglia. There's as many glial cells in total as there are neurons, and they support the neurons. They're important in processing the neurotransmitter. I showed you a very simple recycling mechanism where the neurotransmitter went straight back into the presynaptic terminal. Actually, for glutamate, it goes into the astrocytes, and the astrocytes process it and put it back into the neurons. So yeah, the astrocytes are very important there, and that's something I study in my lab. That's one of the things that we do here in the ANU. Microglia are a little bit like, they're sort of immunology cells of the brain. They move around, they're not connected to any particular brain cell. They move around and they travel around through the brain detecting problems. And when they detect a problem, they get activated, they move to it, and they try and cure the problem in the brain. And there'll be some talks this afternoon, I think you will hear, when we talk about macular degeneration in the eye. You'll hear about microglia, and that's certainly something that in Roxana's lab they study a lot. The capillaries are the blood supply which are going into the brain, and that's very important. It's very important for the oxygen going to the brain. It's very important for removing the waste products and the carbon dioxide. And the neurons really control the blood flow. If you get high activity in a part of the brain, then you increase the blood flow to that area of the brain. And if the blood flow is gradually reduced, this can be one of the triggers for dementia. And of course, if it stops completely, this can be stroke. So obviously that's something also very important, something I'm not particularly studying in my lab at the moment, but there are labs who do study that sort of thing, yes.
Beautiful. Thank you, Brian, so much. Thank you for your talk, and also thanks for answering everyone's question. And thanks everyone for your questions. And if you have more, of course, please email us.